Welcome to this edition of Clinical Clips, an accredited continuing education activity. This brief expert video will spotlight the daily hot topics from the conference.
Hello, I'm Dr. Nikki Mackey, a consultant in HIV medicine based in Imperial College Healthcare NHS Trust in London, UK. I'm also the Vice Chair of the British HIV Association. Welcome to Clinical Clips in HIV. So this is our final summary of the conference data released at EACS 2025 here in Paris, covering October 17th through October 18th. So to start with, I'm sure you're all familiar with the incredible results of the PURPOSE 1 and 2 trials. So yesterday, we learned about persistence to twice-yearly lenacapavir versus oral tenofovir FTC for PrEP in the PURPOSE 2 trial. And what was demonstrated was that persistence was significantly better on lenacapavir, using a strict definition, so that's attending all visits with on-time injections, and tenofovir levels consistent with at least 4 times a week dosing. Adherence to oral TDF FTC decreased over time. Now, the authors did acknowledge that people may have tailored their oral PrEP to times when they felt at risk, so the findings may exaggerate the importance of this difference, but I think we're all very excited about this tool. So continuing the theme of new drugs such as lenacapavir, we also heard the 52-week results from a trial evaluating a complete twice-yearly treatment regimen compared to daily oral antiretroviral therapy. So in this study, people who were suppressed on oral treatment for more than a year with CD4 counts above 200 were randomized to receive the capsid inhibitor lenacapavir alongside the broadly neutralizing antibodies teripalimab and zinlervimab, and from now on I'm going to refer to these as len, tab, and zab, compared to oral treatment. It's important to note that susceptibility to bNAbs needs to be predetermined, and over 200 individuals were screened to achieve the 80 patients that entered the trial. At week 52, 89% of participants received len, tab, and zab remained suppressed, and this was similar to standard of care arm. 3 participants met the confirmed virological rebrand criteria, and reassuringly, all 3 people resuppressed on BF-TAF. So len, tab, and zab were well tolerated, although ISRs were common, and importantly, people preferred len, tab, and zab over daily oral antiretroviral therapy. So the plan is now to move into phase 3 trials with this novel strategy. Another study that we heard about at CROI this year was the EMBRACE study. Now, this evaluated individuals who were virologically suppressed on oral antiretroviral therapy, who were randomized to remain on their oral therapy or switch to one of 2 different formulations of an alternative broadly neutralizing antibody called N6LS, given either intravenously or subcutaneously every 4 months, alongside monthly intramuscular cabotegravir. So here at EACS, we learned about safety and patient-reported tolerability through 6 months. Now, overall, N6LS and cabotegravir were well tolerated, although there were fewer injection site reactions with the intravenous administration of N6LS compared to the subcutaneous formulation. Although all injection site reactions tended to be brief, a few days duration, none actually led to discontinuations. And despite the differences seen with the subcutaneous and intravenous formulations, all participants reported low pain scores and high tolerability, regardless of administration. So based on efficacy, safety, and the positive patient experience, twice-yearly N6LS given intravenously in combination with cabotegravir, long-acting cabotegravir given every 2 months, is being evaluated in part 2 of the EMBRACE study. So we look forward to hearing those results in the future. So although lenacapavir is being evaluated as part of various novel strategies, as we've just heard about, it is of course licensed and available as a treatment for highly treatment-experienced people with HIV, based on data from the Capella study. However, there's few data available regarding the use of lenacapavir in real-world settings, and we learned about the characteristics and virological outcomes of people with HIV initiating lenacapavir-based treatment in a French national retrospective cohort. So this study included 94 people from 13 centres, all of whom were highly treatment experienced, with prior genotypic resistance tests showing drug resistance mutations to at least 1 antiretroviral therapy from 2 major classes. Now, 49 people, that's 52%, were virologically suppressed at the time of lenacapavir initiation, and 48 were viremic. Now, median follow-up on lenacapavir was 13 months. Of those who were virologically suppressed at time of initiation, virological suppression was maintained in 94% of cases. In people with viremia at time of initiation of lenacapavir and optimized background, virological suppression was maintained in 64%, so similar to the results seen in clinical trials. So this study is still ongoing, allowing for increased numbers of participants and longer follow-up. So a study was also presented looking at the effectiveness of fostemsavir in people with 4 drug class resistant HIV. So this was a retrospective study which assessed outcomes in people from the Prestigio registry, which is an Italian cohort. So 52 individuals were included, 42 of whom were viremic at baseline, and 12 were virologically suppressed. Now, they all had 4-class drug resistance. Among viremic participants, 32 of the 40, that's 80%, achieved virological suppression during over a median of 2.3 years. 8 of the 40 individuals who were viremics, that's 20%, and 1 of the 12 who were virologically suppressed experienced virological failure. Overall, 11 of the 52 discontinued fostemsavir with a 4-year probability of fostemsavir maintenance of 70%. Overall, importantly, there was an increase in CD4 count of 34 and also in CD4-CD8 ratio. So that although in many of our settings, numbers of highly treatment-experienced patients, particularly those with viremia, are low, it is reassuring to see that regimens containing fostemsavir can be effective in achieving and maintaining virological suppression. So the combination of lenacapavir, the capsid inhibitor, and islatravir, the nucleoside reverse transcriptase translocation inhibitor, is being developed as a complete weekly oral HIV treatment. Both of these drugs have long half-lives that allow for weekly dosing. At CROI, we learned that weekly oral islatravir and lenacapavir maintained high rates of virological suppression, that's 94%, at week 48 in virologically suppressed people with HIV. And today in Paris, the study team reported on the efficacy and safety of islatravir lenacapavir through 2 years of treatment. So 52 individuals who were suppressed on BF-TAF for at least 6 months with no history of virological failure and no concurrent Hep B infection were randomized to switch to weekly islatravir and lenacapavir or to remain on treatment. And there was then an extension phase at week 48 where patients could roll over and receive the weekly islatravir lenacapavir. No participant had a viral load above 50 at week 96 or at the discontinuation. There might be some concerns with the weekly regimen, but adherence as measured by pill counts was 99.3%, and there was no treatment-related grade 3, 4, or serious adverse events. So this is a novel weekly oral antiretroviral regimen. It's now being evaluated in 2 large phase 3 trials in virologically suppressed patients. This morning, we also saw an elegant study from the Swiss HIV cohort determining the risk of hepatitis B reactivation in people with anti-HBC core positive surface antigen negative individuals with HIV who switched to tenofovir-free antiretroviral treatment. So they identified over 3,000 eligible participants who were core antibody positive surface antigen negative, and of those, 215 were switched to non-Hep B active antiretroviral therapy and 752 to antiretroviral therapy that contained XTC but no tenofovir. Reassuringly, only 3.9% of the participants showed virologic signs of reactivation, so either Hep B DNA or surface antigen positive 1 year after stopping tenofovir, and no participant developed quantifiable Hep B DNA levels. There were no significant dynamics regarding Hep B RNA and anti-core levels after stopping tenofovir, and the study authors concluded that Hep B reactivation does not seem to be of clinical concern in people with HIV with a positive core antibody switching to non-tenofovir antiretroviral therapy. And as this really probably reflects 30 to 40% of our population, this is very reassuring data. Finally, some results from the ACTG 5391 DO-IT trial, which was designed to assess the effect of switching from integrase inhibitor plus TAF-containing regimen to doravirine, a non-nucleoside reverse transcriptase inhibitor, with or without a concomitant switch from TAF to TDF on metabolic health and body weight among adults with HIV and obesity. Now, we previously heard about weight outcomes at IAS this year, and today we heard about the metabolic and body composition outcomes of this trial. So what they found was that a switch to doravirine with or without a change from TAF to TDF did not reduce plasma lipids or insulin resistance at 48 weeks in individuals with obesity. A switch also did not significantly reduce total or truncal fat mass. So I think we need to conclude that larger studies may be needed to clearly delineate the effect of a switch from TAF to TDF on lipids. But this is further evidence, as I alluded to in my previous clip, that our antiretroviral regimen changes are not effective to reduce weight or improve metabolic health amongst people with HIV, and other interventions are likely to be needed. I'd like to thank you for joining us for Clinical Clips in HIV. Please click Continue to complete a brief post-test and evaluation to claim your continuing education credit. Please also return to the activity page to view our first clinical clip from EACS, as well as our homepage for other accredited HIV activities. And I thank you again for your attention.
We hope you found this activity useful and educational. To receive continuing education credit and to download your printable certificate, please return to the activity webpage and select Continue to move through the post-test and evaluation questions.